Okay, so in this lecture we talked about the respiratory system, we talked about the urinary system. So starting with the respiratory system, the big job is to exchange O2 and CO2, and with that, the respiratory system can regulate the pH of blood. We also need it for speech, which is phonation. We filter air as we bring it into the lungs, and we also need it for our sense of smell. So when it comes to the respiratory system, there are four things that have to happen. Okay, these are all known as respiration. The first thing is called pulmonary ventilation. We have to bring air into the lungs and then blow air out of the lungs. So that's just moving air in and out. The next thing is external respiration. So this is the movement of oxygen from the lungs, from the alveoli, into the blood, or CO2 from the blood to the alveoli, to the lungs. Now, once these gases are in the blood, they have to be transported. Okay, if it's oxygen, it has to be transported to the cells. If it's CO2, it has to be transported to the lungs. And then finally, we have internal respiration. This is when you have the movement of oxygen from the blood into the cells, or the movement of CO2 from the cells into the blood. So those are all necessary. So we talked about the functional and briefly about the structural classifications of the respiratory tract. So the functional classifications are the respiratory zone and the conducting zone, and the structural, which I didn't talk about a whole lot, is the upper respiratory tract and the lower respiratory tract. So let's talk about the functional classification. So the respiratory zone, these are anatomical structures that are involved with gas exchange, and there are three of them: respiratory bronchioles, alveolar ducts, and then the alveoli. So here are the respiratory bronchioles, and these respiratory bronchioles will lead into what are known as alveolar ducts. You can't see them because they are surrounded by alveoli, but those are the structures of the respiratory zone. Now the conducting zone, these are just structures that get the air to the respiratory zone. Okay, so things like the nose and the pharynx and larynx, trachea, and pretty much all of the bronchial tree. So there's no gas exchange here. These are structures that get gas or get air to the respiratory zone. Okay, we talked about the pharynx, which has three parts: the nasopharynx, which is going to basically connect the back of the throat to the nose; the oropharynx, which is the back of the throat; and then the laryngopharynx. The larynx is a structure that's going to connect this laryngopharynx with the trachea, and we need it for speech, for phonation, because that's where the vocal cords are. And it also contains the epiglottis, and the epiglottis is going to be like a little valve, like a little switch, that will reroute food to the esophagus instead of the trachea. Okay? The larynx is mostly cartilage. The big ones are the thyroid cartilage, which is the Adam's apple, the cricoid cartilage, and then the epiglottis. And the epiglottis is that little valve that's going to close off the airway. So here we can see some of these structures. So over here on the left, this is the nasopharynx, this is the oropharynx, this is the laryngopharynx down here. So yeah, that's right in succession: nasopharynx, oropharynx, laryngopharynx. And then over here on the right, this is the larynx. This is the thyroid cartilage here. This is the cricoid cartilage. There are other cartilages, like on the back side, that we didn't talk about. And this is the epiglottis, which again is just going to kind of fold over. The opening to the trachea is in here, so it just folds over and makes sure that no food goes down the trachea. So the trachea kind of descends from the larynx, and right at the level of the angle of Louis, or the sternal angle of the sternum, it splits, and it splits into the primary bronchi. So inside there is respiratory epithelium, which is a mucosal layer with ciliated pseudostratified epithelial cells. And there are also goblet cells in there which produce mucus. So the trachea has cartilaginous rings. They are incomplete rings, but the purpose of these rings is to basically keep the trachea open. I talked about the last cartilaginous ring, which is the carina, which is this structure here. And basically it's shaped this way because it allows the trachea to essentially split into the left and right primary bronchi. So the bronchial tree, I'm just going to go to this picture. So the bronchial tree starts with the primary bronchi. So you have one that goes to each lung, And then from there you have secondary bronchi. These are also known as lobar because they go to each lobe. Okay. So on the right we have 3 lobes, so there's 1, 2, and then 3 is down here. And then on the left there's just 2, so 1 and 2. So that's why they're sometimes called lobar bronchi. And then you have tertiary bronchi, which are smaller. Then you have bronchioles. Eventually you get to terminal bronchioles, and then after that you get to the respiratory bronchioles. So it's just like a tree. Everything just kind of gets smaller and smaller and smaller. Now, as we get further down the bronchial tree, these cartilaginous rings actually become plates, and you get less and less cartilage, and you get more and more smooth muscle. Okay, so again, we talked about the respiratory bronchioles, which are the first of those respiratory zone structures, and they lead into alveolar ducts, and off of those alveolar ducts you have alveoli. Alveoli are the gas exchange structures, okay? When, they're going to be arranged in these bundles, these grape-like bundles, those are called alveolar sacs. So again, if we look at this, here are the respiratory bronchioles. This is called a terminal bronchiole. This is a respiratory bronchiole, alveolar ducts in here, and these are alveoli, okay? The whole cluster of alveoli are known as alveolar sacs. Okay, so a little bit of the structure of alveoli. So they are basically one cell layer thick, and most of those cells are called type 1 cells. Those are the cells that gas is going to pass through. On the outside of the alveoli, you have lots and lots and lots of capillaries. Okay, those are the blood vessels that are going to be involved with gas exchange. We talked about the respiratory membrane, which is the combination of the alveolar wall and the capillary wall. Okay, that's what air, that's what gas, oxygen, and CO2 has to pass through. So here we can see how this is set up. Now, some other features of alveoli, they have type 2 cells. Type 2 cells produce detergent known as surfactant, and this decreases surface tension. So basically surface tension is the tendency or the attraction of nearby water molecules to kind of attract each other. And the problem with that inside alveoli is that they can collapse. That makes them want to collapse. So surfactant breaks those forces. Okay, alveoli also have lots and lots of elastic fibers, okay, which makes them a little stretchy. And then we talked about the alveolar pores. So these are the alveolar pores, and the purpose of these is to equalize air. So one alveolus doesn't get more air than another. They also, alveoli have these macrophages known as alveolar dust cells. Here are the type 1 cells. Again, this is where gas exchange is going to occur. And then over here on the right, this is a really good picture. So this is the wall, the type 1 cell of the alveoli. And this is the capillary. This is the endothelial cell. So this is what gas has to pass through. So oxygen is going to go into the blood, CO2 is going to go into the alveolus, and we exhale it. Okay, so the lungs, located in the pleural cavities, which are divisions of a larger cavity known as the thoracic cavity. And we talked about the pleura. So kind of like the heart, the pleura are these serous membranes that line the lungs themselves as well as the pleural cavity. So directly on the lung itself is known as the visceral pleura, and then lining the pleural cavity is the parietal pleura. And in between you have a pleural, it's called the pleural cavity, sometimes called the pleural space. There's fluid in there, and again, the purpose of this is just to reduce friction. So gross anatomy of the lungs, we'll just go to this picture. So the superior portion of the lungs is called the apex. The inferior portion is called the base. Again, the right lung has 3 lobes, so it has a superior, a middle, inferior lobe. The left lung has 2 lobes, a superior and inferior. The left lung also has something known as the cardiac notch, which is going to accommodate the heart. Okay. They are concave on the bottom, so the right lung is concave because the liver sits up under here, and the left one is concave because of both the stomach and the spleen. So again, about the lobes. Okay, then we got into a little bit of physiology. So most of breathing is going to be due to changes in pressure, okay, relative to atmospheric pressure, which is basically the pressure of the gas that you breathe. Intrapulmonary pressure, this is the pressure inside alveoli, and this is going to change as we breathe. But eventually it will equalize with atmospheric pressure, and that'll be kind of the trigger to inhale or exhale. And then intrapleural pressure, this is pressure in the pleural cavity, and this is always less than intrapulmonary pressure. Not only is it always less, it's actually negative. And remember that there are two things that kind of make the lungs want to collapse. The first one is that they are elastic. Okay, so if they stretch, they want to recoil. They want to be their smallest conformation, just like a rubber band. And also that surface tension. That surface tension makes the alveoli want to collapse. We know that surfactant stops that, but another reason that our lungs don't collapse is because of that negative intrapleural pressure. It's like a suction. Okay, there's also surface tension between the visceral and parietal pleura, and that kind of helps the lungs stick to the thoracic cage. Now we talked about if negative pressure equalizes with intrapulmonary pressure, you're absolutely going to have a collapsed lung, no doubt about it. And that's of course known as atelectasis. Air in the pleural cavity is called pneumothorax. So they're related, but they're not the same thing. Okay, so we talked about inspiration. Basically what happens is we increase the volume of the thoracic cavity, and we do that by contracting the diaphragm, which moves inferiorly, and that is going to increase the volume of the chest. You have other muscles like the external intercostal muscles, which also lift the rib cage kind of up and out, again increasing the volume. And what happens when you increase volume is you decrease pressure. And when that happens, that's going to allow air, higher pressure air in the atmosphere, to go into the lungs. Now, if you are, breathing heavily, you might use other musculature to aid in inspiration. So again, this is just showing that. Expiration is mostly passive. So those muscles that we're contracting basically just relax, and that's going to decrease the volume of the thoracic cavity, increase the pressure of the air, and pretty much just force it out. Now again, if you are forcing expirations for whatever reason, you're going to use other musculature. Okay, we talked about respiratory volumes and capacities. So tidal volume, this is just the volume of air that enters and exits the lungs during one breath, and it's about 500 mils. We talked about minute ventilation. Again, I just really want you to know these definitions of volumes and capacities. The inspiratory reserve volume, this is the amount of air that can be forcibly inhaled beyond tidal volume. Expiratory reserve volume, the amount of air that can be forcibly exhaled after tidal expiration. And then residual volume, this is how much air is in the lungs after a forceful exhalation. So the capacities, you get the capacities by combining different volumes. Inspiratory capacity, this is the total volume of air that can be inspired after tidal expiration. The functional residual capacity, this is the volume of air in the lungs after a tidal expiration. Vital capacity, total volume of exchangeable air. Remember, not all of that air in the lungs is exchangeable, because there's a lot of anatomical dead space. Only the stuff that's in the alveoli is exchangeable. So vital capacity is never really close to total lung capacity, which is the sum of all the lung volumes. You've got about 1,200 mLs of dead space in the lungs. Okay, then we talked about the urinary system, which its main job is to eliminate wastes, regulate pH, regulate blood pressure, and obviously it has endocrine functions as well. So the kidneys, we'll just go to the picture. So here are the kidneys. They're paired organs. The right one sits lower than the left one because the liver pushes it down. On the superior poles of the kidneys, you have the adrenal glands. The kidneys are fed by renal arteries, which branch off the aorta. And obviously, they are vital organs. So let's talk about some of the tissue around the kidney, because the kidney is only partially protected by floating ribs, so they need to be well protected, and they really are. So the outermost of this supporting tissue is called the renal fascia, and its job is basically to anchor the kidneys to stuff around it, surrounding structures. Then inside of that you have what's known as the perirenal fat capsule, and this is just a layer of fat, and it's a relatively thick layer. Fat can be a good protective material. And then the fibrous capsule is directly on the kidney itself, and that's why it hurts so bad, when you have any sort of kidney issue, because it's an encapsulated organ, so inflammation hurts. So here you have the renal fascia out here. Inside here is the perirenal fat capsule, and then you have the renal capsule itself on the kidney. Okay, so a little more anatomy. So this is a sagittal section of a kidney. This portion out here, this is called the renal cortex, the outer portion. There are these columns that are cortical tissue that kind of come between these structures, known as renal pyramids. And all these renal pyramids in here, this is known as the medulla. Now these renal pyramids have a little tip here, and this is called the papilla. And this is how the filtrate and eventually urine will flow. So it'll flow down these, and then it will flow into these structures known as minor calyces. Minor calyces drain into major calyces, and then the major calyces will drain into the renal pelvis and into the ureter. So we talked about the renal pelvis, the major calyces, the minor calyces, and inflammation of the renal pelvis, which was this, and the calyces is known as pyelitis. Okay, we talked briefly about the blood supply. As long as you know that the kidneys are fed by the renal arteries, that's pretty much what you need. All right, then we talked about the nephron. So the nephron is the structural and functional unit of the kidney, and it has two parts. It has a glomerulus and a renal tubule. So let's talk about the glomerulus. I'm just going to go to the picture. So the glomerulus is basically a high-pressure capillary bed, okay? And it is fed by an afferent arteriole. So blood comes into this capillary bed through here. And when that blood comes into this glomerulus, it pushes fluid, which we now call filtrate, out into the first part of the renal tubule called Bowman's capsule. Okay, this is Bowman's capsule here. So Bowman's capsule is going to capture this fluid as it's pushed out and then start to drain it down the rest of the renal tubule, which I'll talk about. Now, this capillary bed is also unique because it's drained by an efferent arteriole. So not only is it fed by an arteriole, it's drained by an arteriole too, which is pretty unique. And that'll be more important later. So again, high pressure capillary bed, it's supplied and drained by arterioles. Now the renal tubule has Bowman's capsule, which we just talked about. After that, you have the proximal convoluted tubule, then the loop of Henle, and then the distal convoluted tubule. After that, you have something known as a collecting duct, which is not a part of the nephron. And a reason it's not a part of the nephron is because multiple nephrons drain into collecting ducts. These collecting ducts are actually what are found in those renal pyramids. So here's kind of the overall view of a nephron. Here is the glomerulus. This is Bowman's capsule. Blood comes in, fluid's pushed out, and then it starts to drain through first the proximal convoluted tubule, loop of Henle, distal convoluted tubule, and then the collecting duct. Okay, so the peritubular capillaries. I'm just going to go to the picture again. So these are low pressure capillaries. And what they are going to do, and again, these actually came from that efferent arteriole that drained the glomerulus. They're going to be involved with exchanging things with the renal tubule. Eventually, they're going to drain back into the venous system, so blood can get back into circulation. But that's their main job, is to exchange things with the renal tubule. So you can kind of think of the glomerulus as the first capillary bed. It's high pressure, and this is the second capillary bed. Okay, so basically the glomerulus forms the filtrate, which is in this renal tubule, and the efferent arterioles, or the peritubular capillaries, are going to reclaim that filtrate. Okay, so let's talk a little bit about some physiology. So there are three major processes here. You have glomerular filtration, tubular reabsorption, and then tubular secretion. The filtrate, like I've said, this is going to contain all of the blood plasma components except most proteins. Okay, you really shouldn't have proteins in there. And then urine will have all the waste products, and basically once you get to the collecting duct, it's officially urine. So let's talk about glomerular filtration. So this is a passive process, okay, and it's driven completely by the blood pressure inside the glomerulus. It forces fluid out. Okay, so in that fluid you're going to have water, you're going to have amino acids, you're going to have glucose, you're going to have wastes, and a number of other things too. Proteins are usually too big to pass, and we don't really want them to pass into the filtrate because they're important for keeping fluid in the bloodstream. So the net filtration pressure, these are the forces that basically drive the formation of filtrate at the glomerulus. glomerular hydrostatic pressure, this is the blood pressure in the glomerulus, and it's very, very high for a capillary bed. Okay, and we talked about GFR. It's directly proportional to net filtration pressure. Okay, and I made this little point about if pressure in the glomerulus drops by 18%, filtration will stop completely. Okay, then we talked about tubular reabsorption, and this is basically returning most of that filtrate back to the blood. Okay, so basically these solutes that are in the solutes in water that are in the filtrate are going to move back into the blood. Okay, and we talked about kind of what is reabsorbed where. So remember, reabsorption is when things go from the tubule back into the blood. And here are all of the things and their respective areas. So it looks like a lot, but like I said, you know, learn the exceptions. For example, sodium is reabsorbed everywhere. Okay, glucose is reabsorbed mostly in the PCT. Bicarb is reabsorbed everywhere except the distal convoluted tubule. So learn the exceptions, all right? And again, I'll only really ask one question about it, so just learn those big exceptions and you should be fine. Now, tubular secretion, this is reabsorption in reverse. This is moving things from the capillaries into the filtrate, which will eventually become urine. So this is really important for, you know, removing our nitrogenous wastes like urea and ammonia and drugs and things like that. And we know that we have hormones that stimulate secretion of things like potassium, aldosterone. We talked a little bit about pH. You know, I'm not so concerned about that. Just the big picture here, you know, I want you to understand what reabsorption is and what secretion is. Okay, so urine. So obviously the color of urine depends on a lot of things. The yellow color is due to urobilin, which is a breakdown product of bilirubin. Most of urine, of course, is water. That's going to vary a little bit. The most abundant solute in that water solvent is urea, but there's lots of other things in there also. So the ureters kind of are the continuation of the renal pelvis, and they empty into the urinary bladder. So their main job, and they are muscular tubes, so they undergo peristalsis. They can physically push urine, but they just transport urine to the bladder. And then the urinary bladder, a muscular sac, and its job is to store urine. Okay, the muscle of the bladder is known as the detrusor, and an empty bladder will have these folds called rugae. So here you can see some of this anatomy. So it's really just a storage compartment for urine. Here are the ureters draining into the urinary bladder. So yeah, these are the rugae. So the urethra, you know, its job is to carry urine from the urinary bladder to the outside world. Oops. The rest of this anatomy is not all that important, so don't worry too much about that. And then again, here's the urethra. So as always, if you have any questions, please let me know.